Okay, so we'll start with the megasporogenesis. So what is megasporogenesis? So that is the production of megaspore from megaspore mother cell. Okay, so as we studied the microsporogenesis, so micro represents always the male gametes, megaspore represents the female gametes. So here the production of a megaspore from megaspore mother cell, we call it as megasporogenesis. As we know, so there are nucellus. Okay, so within the ovule what happens? So there is a nutritive portion of a ovule, call it as nucellus. Okay, so nucellus, one of the nucellus cell, they undergo mitotic division and they forms sporogenous cell. Okay, so this is sporogenous, or it is also sometimes called as archisporial cell also. Okay, archi or archisporial, archigonia represent the female structures always. Okay, in the lower plants, antheridia represents the male structures, archigonia represents the female structure. In the same way, so the nucellus, the one of the nucellus cell, they undergo the division and they form sporogenous, or it is also called as archisporial cells. Next, this one of the archisporial cell form megaspore mother cell. So that is our MMC. Okay, so this megaspore mother cell, what happens? They undergo meiosis, that is both meiosis 1 and meiosis 2. Okay, and form what? Again, what is the end result of meiosis? 4 daughter cells. So here also same thing happens. They form 4 cells, and remember this is taking place within the ovule. So I will draw the nucellus portion. Okay, so this is the nucellus. So within the nucellus there will be formation or production of the megaspores. So there will be production of 4 megaspore. Sometimes it is also called as, they are arranged in the linear form and they are 4 together, hence it is called as tetrad. Okay, so this is what the megasporogenesis. So what happens? One of the nucellus, okay, cell or the cell of the nucellus, what happens? They undergo division, they form archisporial cell, or sometimes it is called as sporogenous cell. Okay, from the sporogenous cell there will be production of megaspore mother cell. From where? The megaspore mother cell. And it is important to remember the ploidy also. The megaspore mother cell is a 2n, but all these 4 megaspores which are formed are haploid in nature. This is very important, remember, haploid in nature. Okay, after formation of the 4 megaspore, what happens? Because in angiosperms, the development of megaspore, it is also called as sometimes monosporic development. Monosporic means single spore, but the result is 4 megaspore. But why it is called as still monosporic? Why? Because of within these 4 megaspore, okay, the one, just imagine this is chalajal end, this is micropyle end. Okay, just remember the structure of ovule and imagine this is the chelagel end. Yes, the below end, the above end is a micropyle end. What happens? The megaspore which is present near to the micropyle end, the one megaspore remains active. Okay, one remains active. That is which end? This is very important exam point of view. Micropyle end, the megaspore which is present at the micropyle end, the one megaspore remains active. Remaining 3 get degenerated. The remaining 3 get regenerated and 1 remains active or functional. Okay, so hence the development of a megaspore in angiosperms are also called as monosporic development. So this is what the megasporogenesis. Now we'll go with the development of this one functional megaspore which is developed into mature embryo sac, that is our female gametophyte. Okay, this much you understood, I guess. Okay, anyway, how the megaspores are produced? From the megaspore mother cell, and the megaspore mothers are diploid in nature. They undergo meiosis, that is 1 and 2, and at the result there will be formation of 4 megaspore cells. Within the 4, 1 is functional and remaining 3 get degenerated. Okay, the one which is at the micropyle end remains active or functional. Other 3 gets what? Degenerated. So this kind of development of megaspore, we call it as monosporic development, where only 1 megaspore remains functional, other 3 are degenerated. Okay. So now we'll study the development of female gametophyte. Female gametophyte. So that is the development of embryo sac. Okay. So far now what happens? Within 4 megaspores, 3 get degenerated, and 1 megaspore remains active. This is our single megaspore, and remember this megaspore is haploid. Okay, this megaspore is haploid. Now, how the development of embryo sac takes place? By the single megaspore. This megaspore undergoes repeated mitotic division, and we know when there is division, there is always a 2 steps. One is a karyokinosis, that is nuclear division. Another is cytoplasm division, that is cytokinesis. Okay, so here also same thing happens, but what is the difference is, so when this megaspore undergo repeated mitotic division, beginning they perform only karyokinesis, that is only nucleus division. Later what happens, there will be cytoplasm division and they are differentiated into different cells. But beginning what happens, they undergo repeated mitotic division. Okay, so we can draw like this. The first division, what they form, the 2 nucleus which goes to the different poles or different ends. Okay, this is a stage we call it as 2 nucleated stage. Okay, so next what happens? Again there is mitosis, mitotic division, and they form, each nucleus divides into 2 at the respective ends, and this we call it as 4 nucleated stage. Still there is no cytokinesis, only karyokinesis is taking place. So next is again mitosis. Okay, what they form? How many nucleus it may be? Yes, 8 nucleus. 4, that is we can call it as 8 nuclei. Okay, at the 4 nuclei at the respective places. Now it is called as 8 nucleated stage. Okay, now what happens? Now there is cytokinesis. Cytokinesis, and okay, here what happens in the cytokinesis? 3 cells goes to one end and they form cell wall, cell membrane. Okay, so that is cytokinesis, and another 3 they go to the another end of the yes, they go to the different end. This is our chalagial end and this is micropyle. See, there are 8 nucleus, but here 6 nucleus is divided. So there are remaining 2 nucleus. What happens? They won't get fused, but they stay together and they form this central cell. Okay, so I'll explain you. So like the cytokinesis takes place and the cells are arranged to their respective ends. Okay, so what happens? A single megaspore where 3 get degenerated, a single megaspore undergo repeated mitotic division, 2-celled, 4-celled. When they come to the 8 nucleated cell stage, what happens? The cytokinesis takes place, that is cytoplasm division. So the 3 cells will arranged at the chalagial end, remaining 3 arranged at the micropyle end, and the center 2 nuclei, what they do? They form polar nuclei or secondary nucleus and they form a central cell. This is one big cell, central cell. 1, 2, 3 at the chalagial, 4, 5, 6 at the what? Micropyle, and central 1 cell. Now this, how many cells now? Yes, 7 celled. How many nucleus? 8 nucleated stage. This is what we call it as. Okay, so this is what the structure of embryo sac or the matured female gametophyte. As we studied, after the cytokinesis, what happens? Those 8 nuclei is distributed into the different cells. Okay, the 3 cell after the cytoplasm, what happens? Those 3 cells will go and remain in the chalagial end. Those 3 cells are called as antipodal cells. Okay, antipodal cells, and they are always present in the chalagial end. Next, remaining 3 will go and stay in the micropylar end, that we togetherly called as egg apparatus, which consists of the egg cell and synergids. And In a synergids, there will be the cellular thickening of synergids itself, which look like finger-like projections within the synergids. These finger-like projections is called as filiform apparatus. Okay, so there are, as I said, 7 cells. 1, 2, 3, 4, 5, 6 cells, and the remaining cell is called as central cell. This itself a central cell, where the 2 remaining nuclei are present. Okay, how they are present? They are not present in the fused state. They are together. Okay, and that we call it as a polar nuclei, or it is also called as secondary nucleus. Okay, now it's time to study the ploidy, because it is very important when we study the fertilization process. So it is very important to study what the ploidy, what is haploid and diploid cells which is present in embryo sac. As we know, our megaspore, which is single megaspore, which is functional, undergone the development of the female gametophyte, are haploid in nature. So hence the formed embryo sac is also haploid in nature. So all these antipodal cells are haploid. Okay, and this egg apparatus consisting of egg cell and our 2 synergids are also haploid. Okay, and here the polar nuclei or the secondary nucleus, what happens? As there are 2 nuclei which are together, hence N and N, it makes 2N. So except the secondary nucleus, all other cells are haploid in nature. It is very important question. They may ask the ploidy of all these cells. Okay, which is very important. Understood? Okay, so next, what happens? How, as I said you early, the pollen tube, which is formed by the tube cell, which in turn carries the male gametes, okay, which is formed from generative cell. Remember the previous sessions. Okay, that is carried by the pollen tube, and it should get fused with the egg cell, but they are present within the embryo sac. And as I they go into the embryo sac from the end of micropyle end. How they will get to know? They should enter from the micropyle end. So the filiform apparatus, which is present in the synergids, will show a route to the coming pollen tube to get entered into the embryo sac. Then it get fused with the egg cell to perform the fertilization. Understood? And this structure is very important. The embryo sac structure, they may ask to draw a neat label diagram of embryo sac. Sometimes female gametophyte also they may ask. Both are similar, female gametophyte and embryo sac. And next question is draw a neat label diagram of anatropous ovule. And one thing I need to tell you, that is, when we draw the anatropous ovule, I have labeled only embryo sac there. Okay, now you will now you know the different cells which is present in the embryo sac. Please don't label only embryo sac in the anatropous ovule. Instead of embryo sac, label all these different cells, that is antipodal cells, secondary nucleus, central cell, egg cell, synergids, egg apparatus, chalical and micropyle and everything. Okay, please don't forget and practice these diagrams, which is very important. So in the next class, we have finished the one event in the pre-fertilization event. So next sub event in the pre-fertilization event is gamete transfer, and in plants the gamete transfer is of pollination. Yes, the gamete transfer in plants is by the process called as pollination. We are going to study the definition, different types, different pollinating agents, fascinating information we are going to study about the pollination. Okay, thank you.